All right, the third type of carbon molecule that we need to talk about is proteins. Proteins are the structural material of the body. All right, and they too have monomers, just like all the other ones. There's the other two, the fats and the carbohydrates. So the monomers for proteins are called amino acids. And amino acids have a specific structure. So an amino acid looks like this. So it starts with a carbon molecule. So it's right here in the middle. And an amino acid has sort of an acid group, and then it also has this weird functional group off it called an amino or an amine group. So it has, let me get my color. It has an acid group on one side, and on the other side, it has a nitrogen group. So an amine group, which just means it has a nitrogen in it. And on this side of it is just a hydrogen. Okay, so there's 2, 4, 6 bonds there. And then on this other side, in green, is what we call the R group. So the R group is just any molecule that is connected to it that makes the amino acid what it is. And what I mean by that is there are 20 named amino acids. About 10 of them are non-essential, and 10 are essential. So what do we mean by that? Well, essential means you have to get it in your diet. Non-essential, your body can make it, okay? But this part right here, this R group here, whatever it is, it could be an oxygen and a hydrogen or whatever it is over there. That's what makes it what it is. All amino acids have this structure. Let's put it in red here. So all aminos have this. And where they are different is in the R group. So amino acids are linked together to form proteins, okay? And when that happens, they link with their nitrogen to a carbon, okay? So if we have, we have this amino acid here. Carbon. I'm trying to make my colors, but it's kind of taking me longer. So carbon here. And we're going to connect it to an amine. And my color is not right, and I apologize. Nitrogen, hydrogen there. Carbon, and here's my acid from the other carbon. It should be blue, but I'm sorry. Okay, so here are the R groups. So this bond right here, I'll just put that in red. This bond here, which didn't turn red, why is it not turning red? So this bond here is what bonds the two amino acids. This is amino acid over here, and here's an amino acid over here. And it's bonded here at this bond. And this bond, which we get through dehydration synthesis, just like everything else, that's called a peptide bond. So when we link two of these amino acids together, we get a dipeptide. If we link another one to it, that would be a tripeptide. If we have a chain of amino acids less than 100, so if we have a chain of amino acids that's less than 100 amino acids long, then we have a polypeptide. If it is greater than 100 amino acids long, it's officially a protein. All right, so proteins come in different shapes, okay? When they are formed, we link them together in like a line. So this one's linked, this one's linked to this one, and we put another one, link it to that one, and we make a big line of proteins. That structure of a protein is called a primary structure. Oops, sorry guys. Just put this over there. Okay, so that's a primary structure. If we take that strand, so that line of amino acids, and we can either curl it around like this in sort of a helical configuration, that is called an alpha helix. And that's what's called a secondary structure. Okay? We can also form them into sheets. And this would be called a beta sheet. Both of these are secondary structures of a protein. The third way is called a tertiary structure. And the tertiary structure is when we take an alpha helix or a beta sheet and intertwine them together and bend those around. Okay, so the alpha helix bends it around, or the beta sheet, we bend it all around. Okay, that structure is a tertiary structure. There's also a quaternary structure. And that's where we take a three-dimensional alpha helix tertiary structure or a beta sheet tertiary structure, one that's already been bent all around, and we put those together. Okay, so two or more of those polypeptides that are already in that tertiary structure, we combine together, and that makes a quaternary structure. An example of that would be hemoglobin is that way. So hemoglobin has 4 different proteins attached to one another, just like this. okay? The structure, this is important because the structure is very important. It's important for the function of a protein. If a protein is denatured and it loses its structure, it can't do its job. It can't do what it's made to do. So the structure is very, very important. All right, so just remember, proteins made up of amino acids. Amino acids are the monomers. Okay, amine means it's got a nitrogen. Acid means it's got an acid group on it. Amino acid. And links of those make polypeptides and proteins.